So good morning everyone. We will start with the new topic, photorespiration. So in the previous topic we have studied the C3 cycle. So this photorespiration also takes place only in the C3 plants. C3 plants. So what happens now when we studied about RUBP? RUBP. So in that we studied about an enzyme Rubisco, which has affinity for both oxygen as well as carbon dioxide. So like when the carbon dioxide is there in the atmosphere, it binds with RUBP and we will get 3 PGA, that is in the C3 cycle we studied. But now what happened? What will happen when the carbon dioxide concentration is very low? It is very low and oxygen is very high. So as the name suggests, Rubisco, ribulose, bisphosphate, bis, carboxylase, oxygenase. So it has affinity for both carbon dioxide and oxygen. So now oxygen is high over here. So it will RUBP. This will bind with oxygen. And produce two compounds. So it is a 5 carbon compound. So it will produce two compounds. So which are these? First is that only 3 PGA. 3 PGA. What is that? 3 phosphoglycerate or glyceric acid. And one will be phosphoglycolate. So you can guess this is a 3 carbon compound. So how many carbon compound will be this? So 5 carbons are there. 3 carbons over here. 5 minus 3, 2. So it is a 2 carbon compound. So what will happen now? You know that this reaction is taking place in the chloroplast. I will write here chloroplast. And here what will happen? That phosphoglycolate will be converted into glycolate. So you can see that the phospho part of the glycolate is removed. So what we can write is that inorganic phosphate is removed. So this glycolate I am making a box over here. So this glycolate, keep in mind. So now what will happen? This glycolate will be transferred into peroxisomes. So what is transferred? The glycolate. So what will happen to this glycolate is that it will readily combine with oxygen and give glycoxylate. So this glycoxylate will get converted into, so the H2O2 that is peroxide, that is removed out of the cell because it is very dangerous to the cell. As in the cell chapter we have studied that peroxisomes have the function that they convert this peroxide into water, because peroxide is very destructive for the cell. So this is gone out. So now what will happen? By transamination, we will get amino acid glycerine. The process is transamination because it is because this produced is amino acid. So here amination is here used. So glycine is produced. Now this glycine, remember that we have to use the cycle from chloroplast and peroxisome twice because we need 2 glycine in the next step. So be careful. Here I am drawing another organelle, mitochondria. Mitochondria. So what will happen? These two which are there na, glycine, are getting converted into one serine. Acha, these are converting into one serine. So what will happen? Keep in mind, the carbon dioxide is lost. The NH3, that is the amine group is lost. And what will happen is that the NAD plus will be converted into NADH will be converted into NADH. So here you are remembering that it is respiration, so we are using NAD plus over here. It's getting converted into NADH. So now there will be a reverse process. So box the serine. So what will happen now? See, box the serine. So I am drawing now all the 3 organelles in line. So down here it is the mitochondria, here is the peroxisome, and here is the chloroplast. I am denoting it short. Chloro and mito. So we got here serine. So this serine will jump into this where peroxisome, and this serine will be converted into, see over here, will be converted into dehydroxypyruvate. And this will get converted into, and this will get converted into glycerate. Now what will happen? The NADH which was formed will again get converted into NAD plus. So this glycerate will jump into the chloroplast, and glycerate will be converted into 1 PGA. So glycerate will be converted into 1 PGA by what will happen, that ATP, here it is phosphoglycerate, right? So I have to break the ATP. ATP will be broken down into ADP plus 1 inorganic phosphate. So PGA are formed. So now you can see that first in the starting process 1 PGA was forming. So by that we are getting that 2 PGA are produced. But now what will happen, that I will tell you that how this process is unleashed. See here carefully. In C3 there were 4 PGA overall. If 2 cycles were there were 4 PGA. But in C3's photorespiration, there are only 3 PGA for 2 turns. So what will happen now? 1 is lost. So 1 carbon dioxide was released in the mitochondria. 1 carbon dioxide was released. So 1 carbon dioxide. So out of 4 carbon dioxide here are fixed. Okay. And here maybe there are 3 fixed because 1 is lost. So now 3 by 4 of 100%. So what I will get? 25, 75. So 75, 100 minus 75. So 25% carbon dioxide is lost. 25% carbon dioxide is lost. Because if here 4 carbon dioxide are fixed and here only 3 will be fixed, because this and this is directly proportional. This and this is directly proportional. So this is because NCRT calls is a waste reaction. Why? Because when glycerate is converting into 3 PGA, the ATP is used but no energy is produced, as the carbon dioxide should be fixed but it is released in the mitochondria. So this photorespiration is considered as a waste reaction, So let us recall now what we have studied. So just take a blank page and draw this diagram. I am giving you space to draw. So first you have to draw like this boxes you have to draw. So this is of chloroplast, this is of peroxisome, this big is of mitochondria. So maybe there may be not much space, but you should be able to draw that. I am writing short RU, maybe you can zoom the screen. RUBP plus O2 gives what? 3 PGA. Now recall. I am writing every step in detail. 3 PGA plus what? Phosphoglycolate. So now phosphoglycolate will be getting converted into glycolate, right? So this glycolate will enter what? What is this? Recall fast. What is this? Peroxisome. So glycolate entering peroxisome, this will readily pick up oxygen and get converted into glycoxylate, and this glycoxylate, by which process it will be converted into glycerine? Recall transamination. Yes, right. Transamination into glycerine. So this is glycerine. This will enter. So how many glycerine will enter fast? For one saccharine, how many glycerine will enter? Yes, it is correct. 2 glycerine will enter. Now this will get converted into which amino acid? Yes, good. Serine. And what will happen here? Carbon dioxide out. Okay, good. NH3, the ammonia, or you can call it as ammonia, that will go out, and NAD plus will be get converted into NADH. So serine will go up. Serine will get converted into dehydroxy pyruvate, and dehydroxypyruvate will be converted into glycerate. What will happen over here? Recall. Yes, NADH will break to give NAD plus, right? And this glycerate will again come in the chloroplast, and break down into 3 PGAL. So by which process that ATP is utilized, because I need one phosphate here. ATP is utilized into ADP plus 1 inorganic phosphate, which I utilize here. So for glycerine we are this is into twice we have to do. So this formed will be how much? Tell fast. This formed will be how much? 2 PGA will be formed, right? This will be 2 PGA will be formed over here. And what will happen? By this 2 PGA here it will be formed, and by this 1 PGA over here. So total 3 PGA. If there will be a question how many PGA are forming, so 3 PGA are forming, and these are directly going into where? C3. Where these are going directly in the C3 cycle. So they will ask a question, how much percentage of carbon dioxide is lost? So how much percentage of carbon dioxide is lost? That is 25% of carbon dioxide is lost. So I am naming the organelles over here so that you can take a screenshot or right. Chloroplast, peroxisomes and mitochondria. So one more question can be framed, that when does this takes place? This takes place when the oxygen is high, the carbon dioxide is low, there is high temperature, and we can call it a stress condition when the water is low. This is the photorespiration. This all things are not in the NCERT, but for you to understand in detail, this all is taken and why it is a waste reaction, because carbon dioxide is released, because but it should be fixed, and here the ATP is consumed. ATP consumption is there, but there is no production. So this is all about the photorespiration. You can revise this again and draw this diagram. Thank you.